Chapter 5 is our last chapter of transmission genetics before we jump into Chapter 25, which is population genetics. So this chapter really looks at the exceptions to Mendel's original rules that we discussed in Chapter 3 and then expanded on in Chapter 4. So the first principles that are listed here in red are things that we've already talked about. So we've talked about sex-linked traits, we talked about penetrance and expressivity, as well as incomplete dominance. What Chapter 5 focuses on are exceptions to those Mendelian principles. So today we're going to concentrate on the rest of this list and will all the way through Chapter 5. So one place to start is just to review the definitions of the different types of dominance. So in Chapter 3, we talked a lot about complete dominance, which is where you have a phenotype that of a heterozygote that is the same as the phenotype of the homozygote. And so that's our dominance recessive relationship. Incomplete dominance is where the heterozygote phenotype is intermediate, so it falls within a range between the two phenotypes that are expected. And then codominance, we spent a little bit of time on in Chapter 3, but are really going to look more deeply at it in Chapter 5. And that's where the phenotype of the heterozygote includes both the phenotypes of the homozygotes. So one of the first principles that really deviates from Mendelian principles is epistasis. And epistasis is a masking effect where one gene actually masks another. It works similarly to dominance, except that there is no common loci among alleles. So the gene that actually does the masking is called the epistatic gene, whereas the gene that is being masked is called the hypostatic gene. And the best example that we have of epistasis is actually Labrador retrievers, to where you can actually get blocking of genes that will result in either a yellow, a black, or a chocolate lab, depending on which genes are blocked. So some of the different types of ratios that we can observe with epistasis are shown in table 5.2 here. Our traditional 9:3:3:1 ratio doesn't have any type of interaction associated with it. However, if you start to see ratios that are a little bit odd from that original 9:3:3:1, you know that you're getting into some sort of epistatic pattern. So spend some time on this table and just realize that these modified gene results are because of an interaction between one or two different genes, and sometimes it's even up to 5 or 6 genes. So coat color genetics is really complicated. We're going to briefly just talk about genetics of coat color in all other species except horses. Horses are their own ball of wax, and I'm going to do a supplemental lecture for horse coat color if you guys are interested. So when we think about general coat color, like in dogs, there's really 7 different loci or 7 different genes that are going to affect the coat color display pattern in the phenotype. So those different those different genes are shown down at the bottom of this slide, and just realize that for every gene they're going to have multiple alleles. So the process of coat color determination in offspring becomes a pretty fine art in a science. Some of the different color patterns that we see in different breeds of dogs are actually shown in their corresponding genotypes within the breed and then across breeds as well. So if we look at a pretty complicated one like the beagle up here, they're going to have 3 different genes that interact to form their specific color pattern. Rottweilers also have a large display. They have 4 different genes that are going to affect their coat pattern, whereas poodles and greyhounds are very simplistic, so they only have 1 gene that really affects their phenotypic color. Some of the other modifications or the deviations from Mendelian inheritance that we see has to do with sex and the influence of sex on heredity. So we talked a lot about sex determination and sex linked characteristics in chapter 4, but here in chapter 5 we're really talking about how sex can either mask or enable a phenotype to be expressed, and the expressivity of that expression pattern. So some of the sex influence characteristics that we'll be covering in chapter 5 refers to beard development in goats, as well as male pattern baldness in heredity from generation to generation. And then we'll also be talking about sex limited characteristics, where we start to see different types of phenotypes or variations within one phenotype for a specific trait having variable expressivity. And one example of that is cock feathering in poultry. And then finally, sex limited traits. The main one that we're going to talk about is precocious puberty. So, precocious puberty really doesn't start to display itself until the individual starts to go through the pubertal process. And precocious means too soon. So, that means that puberty is actually happening in young girls and young boys around ages 7, 8, and 9. One other type of inheritance pattern that deviates from Mendelian principles is cytoplasmic inheritance. And this is a big deviation because we're not talking about the genomic material that's contained in the nucleus. We're actually talking about cytoplasmic organelles, so the mitochondrial effects. So we do see certain organelles that have their own genome in the cell. So this is extra of the nucleus. And this is mostly mitochondria and some of the chloroplasts. In the mitochondria, there's approximately 35 to 37 genes that encode that DNA. And that's very, very small compared to the nuclear chromosomes. However, cytoplasmic inheritance becomes a big deal because during segregation, the mitochondria is going to segregate randomly during cell division. So not all of the genetic material is actually going to divide into those two new cells evenly, and it's definitely not going to do it as part of a system. So you can actually see phenotypes reflecting different types of mitochondrial DNA because of this non-systematic division. Some of the genetic maternal effects that we're going to talk about in chapter 5 really have to do with seashells, or snail shells, and the actual sex of the offspring can affect what type of coil that the shell actually has. So a dextral coil is going to coil to the right, central is going to coil to the left, and the mother's genotype is actually going to determine the offspring's phenotype. So the genetic maternal effects is where we see an offspring phenotype for a specific trait that is not determined by its own genotype. So its own genome is not dictating what is actually being expressed, but it's actually going to be dictated by what the mother was. Another concept that we're going to talk about is genomic imprinting. And imprinting is where you have genes that are on autosomes, both in male and females, and that are going to make up a genetic contribution to a new organism. Usually we assume that the contribution from mom and dad on these autosomes is going to be equal, but that's not always true. So some genes are only going to be expressed if the male is a parent or if the male is a female, and vice versa as well. So some examples of these type of conditions actually are pretty serious. So Prader-Willi syndrome, Engelmann syndrome, and then bipolar disease as well as carcinogenesis are really genomic imprinting conditions. And carcinogenesis is probably the one you guys can relate to the most. So we've heard for a long time that that smoking while pregnant really puts the the baby at danger for developing carcinogens or cancerous tumors in their system. So even though the baby didn't have any control over that action, the mother did. She's actually influenced the ability of that baby to survive and express a correct phenotype that's going to let them survive throughout their entire life. And finally, anticipation is one of the last conditions or deviations that we're going to talk about. An example of anticipation is myotonic dystrophy, and this is where you have some sort of condition like MD or Graves or Crohn's disease that actually gets worse with each generation. So, for example, I have relatives that have Crohn's disease, and even though their symptoms, the parent system symptoms are pretty minor, their offspring potentially could have very severe Crohn's disease symptoms as well. So those symptoms get worse from generation to generation. So they anticipate or are exacerbated each generation. And then finally, we're also going to talk about some environmental interactions. So temperature is an easy one to relate to. We see it in marigolds, so we can actually see different growth depending on what degree the plant is actually housed at. And then in rabbits as well, we can see a Himalayan versus a full white rabbit. If those rabbits are housed at 20 degrees or less, they'll have black points. If they're reared above 30 degrees Celsius, then they'll be solid white as well. So interesting color patterns just because of the environment. So that really sums up what we're going to talk about in Chapter 5. Again, it's a lot of material, a lot of different types of exceptions to Mendel's principles. So I encourage you guys all to start making a list and making examples and getting a good handle of what's going on. This is an excellent, excellent place for flashcards. All right, we will see you guys in lecture.